If you take a human body and break it down to its smallest physical components, you are left with an assortment of carbon, hydrogen, and oxygen. On their own, these atoms and molecules are completely lifeless. So where exactly does life begin? 3.5 billion years ago, Earth was a hostile place. In environments similar to the modern Puga Valley, chaotic chemical reactions happened constantly. For life to emerge, something had to separate delicate molecules from the violent world outside. That separation requires a distinct physical boundary. Humanity first began to see evidence of this in 1665, when researchers started using early microscopes to examine biological structures. Looking at a piece of tree bark, scientist Robert Hooke saw thousands of tiny, empty compartments he called cells. But these were just the abandoned boundaries of dead matter. It wasn't until Antony von Leeuwenhoek examined a drop of pond water that we witnessed living cells, swimming and actively surviving within those borders. These early observations pointed to a necessary condition for survival: isolation. To stay alive, an organism must maintain a physical barrier that prevents its internal chemistry from being overwhelmed by the external universe. In many organisms, the first layer of defense is the cell wall. Plants build theirs out of cellulose, while bacteria use a sugar-protein web called peptidoglycan. This rigid barricade protects the cell from mechanical damage and locks it into a stable shape. But every living cell, regardless of whether it has a stiff outer wall, relies on a much more sophisticated inner boundary: the plasma membrane. This diagram illustrates the fluid mosaic model, the core structure of the membrane. It is built from a double layer of lipids. The water-loving heads face outward toward the environment, while the water-fearing tails hide in the middle. Proteins are wedged throughout this lipid sandwich, like tiles in a mosaic. This structure is constantly flowing. The lipids and proteins aren't locked in place. They drift and ripple alongside each other, behaving more like a dense liquid than a solid wall. This fluidity allows the membrane to be selectively permeable. It acts as a mechanical filter, opening and closing to dictate exactly which materials are allowed to enter or exit. When materials need to cross this border, the cell often relies on passive transport. This costs the cell zero energy. Instead, it runs on natural physics. Molecules will automatically spread from an area where they are highly concentrated to an area where they are scarce. We call this movement diffusion. If a cell uses up its internal oxygen, the density of oxygen inside drops. The high concentration oxygen outside naturally slips through the membrane to fill the void. Simultaneously, carbon dioxide built up inside diffuses outward into the emptier environment. When this process involves the movement of water, it is called osmosis. Water molecules slip through the selectively permeable membrane to dilute areas where sugars or salts are heavily concentrated. This physical pressure can drastically alter the cell. Place a cell in a hypotonic solution, water with very few dissolved particles, and water rushes inside, causing the cell to swell. Drop it into a hypertonic solution packed with salt, and the cell's internal water rushes outward, leaving it shriveled. While passive transport is efficient, it is entirely reactive. Without spending energy, the cell is at the mercy of the natural physics of its environment. Survival often requires fighting against the environment. A plant may need to extract scarce nutrients from depleted soil, or a cell might need to push waste out into an area that is already crowded with debris. To solve this, the cell uses active transport. This process forces molecules to move in the opposite direction of their natural flow, moving from low concentration into high concentration. Moving against the gradient requires physical labor. To do it, the cell must spend its internal energy currency, a molecule known as ATP. By spending ATP, a cell can physically reshape its border. During endocytosis, a single-celled amoeba extends its membrane to surround food particles, eventually engulfing them and dragging them inside. The cell also uses ATP for exocytosis. It packs waste material into small bubbles that merge with the outer membrane, forcefully expelling the contents back out through the barrier. This ability to burn energy to defy natural physics is what makes a cell an autonomous living machine. It allows biology to maintain its own internal order, regardless of the chaos outside. Because the membrane regulates the border so strictly, the cell interior becomes a protected environment. This internal living matter, called the protoplasm, is commanded by the nucleus and kept safe from external interference. Within this sanctuary, the cell can grow, process energy, and eventually replicate its entire architecture to create a perfect copy of itself. As these cells multiply by the billions, they link together to form tissues, organs, and eventually the vastly different organisms we see in the world today. This blueprint holds true across wildly different scales. An unfertilized ostrich egg is just one massive cell, holding a vast reservoir of nutrients within its boundary. Compare that giant egg to the microscopic neurons in a human brain. One is a storage vessel, the other is a branching communication network, but both rely on the exact same membrane physics to survive. The cell is life's fundamental unit because it is the smallest entity capable of drawing a line in the sand. By regulating its borders, it sustains the delicate internal balance required for life to exist in a lifeless universe.
Thank you so much for watching. I'll see you in the next one.